Well, good evening. My name is Ruth Ballard. I'm the chair of Biological Sciences at Sacramento State, and I want to welcome you here to our university, to our Topics in Regenerative Medicine series. This talk is the third lecture of a four-part series that we host each academic year. In addition to graduate and undergraduate students, the audience includes science professionals and members of the general public interested in stem cell and regenerative medicine. Our final seminar will be on April 21st, and our entire seminar schedule can be viewed on Sacramento State's Science, Technology, Engineering, and Mathematics, quote unquote, STEM website, which is not the same thing as stem cells. It's very confusing. This lecture series is co-sponsored by the Center for STEM Excellence, which promotes the advancement of STEM disciplines. The California Institute of Regenerative Medicine funds our MA Professional Stem Cell graduate program in the Biological Sciences Department through a Bridges to Stem Cell Research grant, which is a collaboration between UC Davis's stem cell program and the College of Natural Sciences and Mathematics here at Sacramento State. The department is currently reviewing applicants for our next year's professional MA stem cell program. We're very excited to announce that our fifth-year cohort of stem cell master's students will graduate this coming May, and cohort 6 members are actively working on research projects with various UC Davis investigators. If you're interested in our program, please contact Dr. Rosalie Sprowls in the Department of Biological Sciences at Sacramento State. In preparation for this evening's talk, please take a minute to silence your cell phones. Our speaker will not be accepting questions during his talk, but will open things up to the audience at the end, handing around a portable microphone so everyone can be heard. It's a large room. Now that the preliminaries are out of the way, I'd like to introduce Dr. Bauer, who will give us a 5-year update on stem cell and gene therapy product manufacturing at the UC Davis Good Manufacturing Practice, GMP, facility. Dr. Bauer is the founder and director of this facility, where a number of researchers have been producing, under strict regulatory oversight, novel clinical-grade stem cell and gene therapy products. The translation into clinical applications to treat patients suffering from illnesses has provided valuable options that would not have existed without the resilient work of these dedicated teams of professionals. Highlights among many other products manufactured are gene-corrected skin cells derived from patients' own induced pluripotent stem cells for the treatment of a devastating skin blistering disease and a stem cell treatment for blindness. Gerhard Bauer has authored numerous articles, publications, and has designed and directed several GMP facilities. The UC Davis GMP facility is currently the largest academic GMP facility in Northern California. Our professional master's students benefit from this training at this truly unique facility. Dr. Bauer continues to mentor students from various academic institutions and has served as mentor for several of our graduate students. Please join me in thanking Dr. Bauer for taking time out of his busy schedule to be here this evening and in welcoming him as our honored guest.
Thank you very much. This is what a wonderful audience tonight. So many people. Thank you all for coming in spite of St. Patrick's Day. But after the lecture, then you can go out and you do your St. Patrick's Day thing at De Vere's. Okay? So, 5 years. The GMP facility has been up and running for 5 years already. Well, I thought it was just yesterday that we moved in. Let's have a look what we actually accomplished in the GMP facility at UC Davis. Ha! This is what people want from me every day. Hey, cutting-edge treatments are available. Let's get them now. Okay, I can do it like right now. And so, hold on a second, okay? Because there, I'm a history buff, and I want to show you a little bit about history. In 1901, diphtheria patients. You know what diphtheria is? It used to be a terrible childhood disease. Nowadays we have a vaccine against that. And it's bacteria that colonize your airways, and they make membranes. It's pretty bad, and inflammation goes on, and a lot of people die from that. So diphtheria patients were routinely treated with antitoxins derived from the blood of horses. And why? Well, you don't really die from the bacterial infection itself. You die from the toxic effects, the toxins that the bacteria make. So people thought, if we neutralize the toxins, maybe we can help these patients, and that's exactly what they did. They didn't know anything about antibodies, but they knew if they immunized a horse and took the blood from the horse, actually the serum from the blood of the horse, and injected that into patients, it would neutralize the toxin, and patients had a chance to survive. However, at that time, there were no centralized or uniform controls in place, and the antitoxin was made in local manufacturing plants. And you know what local manufacturing plants are? It's the stable behind the house where the horse was standing. So in St. Louis, of all places, again, you know that combination had tragic consequences. 13 children died after being treated with diphtheria antitoxin made from the blood serum of a tetanus-infected milk wagon horse by the name of Jim. And this was followed by a similar tragedy in New Jersey that involved deaths and injuries of more children. And then the government stepped in because this could not continue like that. And Congress enacted the Biologics Control Act of 1902. So July 1st, 2015 will mark the 113th anniversary of this law, which gives the Center for Biologics Evaluation and Research, CBER, at the FDA, authority to regulate biological products and ensure their safety. So it's that long ago, because they realized that long ago that we have to do this manufacturing of products safely, that they started to enact this law. And in this particular law, the first word, like good manufacturing, was mentioned. So good manufacturing practice came out of that. And this is what we're still doing. And now you know where this comes from. And if you put a little C in front of GMP, a lot of people may have seen that. So CGMP. That means current good manufacturing practice, because we don't want to do the same manufacturing practice as we did in 1902. So it evolves with science. Now, our goal always is improve patient care through cutting-edge research, but most importantly, do no harm. Have you had the doctor do harm to you? Perhaps, because they didn't know exactly what they were doing. This has happened. And we want to make sure that we don't do any harm to our patients, but we really make sure that we know exactly what we're putting into the patient, that we evaluate all the parameters of the product beforehand, as good as we are able. And for that, this particular slide I have been showing now for many years, and this, I think, is really what everything revolves around. It's how we are setting up research from an idea to the patient. We do this in the following way. We do basic research. A lot of our students here are doing basic research in the laboratory. They want to find out how certain molecules work, what their effect is on cells, what their metabolism is, and so on. Well, after you have worked all that out and you have an idea and say, this could actually be used for the treatment of a certain disease, we then go into translational research. We try in the appropriate in vitro and in vivo model how this will be applied to cure, possibly cure, a disease. And for that, we have cell lines in the laboratory that we can treat with these products. We also have other tests in the laboratory that measure certain parameters, biochemical parameters, but most importantly, the FDA requires, and this is not, and if we want to do it, but this is a mandate, an in vivo model where we can mimic the disease, and then a model that allows us to treat the disease in a similar way as we would treat it in the patient, in the human patient. And luckily, mice are our most important animals, and we have a lot of diseases that we can mimic in the mouse and then can translate into a research product that may benefit humans. Finally, after that is accomplished, we go into the GMP facility, and there we make the product under good manufacturing practice conditions that are current. And finally, we end up with the product in a little syringe that goes into the patient. This is a historic slide. I want to show you something. Here, this incubator here. You see that on the left side? It contains all of these flasks. This is how we did our first stem cell gene therapy for HIV clinical trial in Los Angeles in 1997. So this is how we did it. And this is the syringe that we used to inject the patient with. And the patient is still all in order. There is no adverse event from that clinical trial, and we have many, many years of follow-up now, and we can say that stem cell gene therapy for this application was safe. So, what is required to bring a novel cellular product to patients? As you heard first, good manufacturing practice, but what is it? What is the law? What does it say? It's a national standard for the production of pharmaceuticals that assure safe and effective drugs. And it's written down in 21 Code of Federal Regulations, 210 and 211. I rattle this down like that because I've been dealing with this for 20-something years, you know? This is what I tell people all the time. Look it up, it's in 21 CFR, and stick to it. But there's a second aspect to that. As soon as you come to a patient, you need to make sure that the patient is treated well, and that is good clinical practice. It's an international, ethical, and scientific quality standard for clinical trials. We want to make sure that patients that participate in a human clinical trial are protected, that their rights and safety and well-being is respected, and that clinical trials are credible. But do you see the two little things here that are different? What is GMP? It's national. And what is GCP? It's international. So if I make a product here in my GMP facility, can I send it to Europe to be used in a clinical trial? No, I can't, because they have different GMP rules, and I must comply with their GMPs to also participate in a clinical trial with products that I manufacture. So it's very important to remember. And it works the same way when the Europeans want to send us GMP material. So, having these two things in mind, let's move forward. These are the products that we're manufacturing currently after 5 years in the UC Davis GMP facility. It's 16 different things. I'm not going to talk about everything, but you're supposed to memorize everything, and afterwards it's a test before you can get out. Okay? So let me give you a little overview of what we're doing here. We're taking stem cells, autologous, from the patient, from the bone marrow. We're taking also mesenchymal stem cells. We're using CD34 positive hematopoietic stem cells. We're making retinal progenitor cells for patients with blindness from donor tissue. We're making corneal cells for corneal repair. We're making even microorgans that we can implant into a person and that secretes a certain factor over a prolonged period of time to replace the drugs that you would take every day. You don't have to take them because the microorgan makes them. We have used human embryonic stem cells to make neuronal stem cells for the treatment of ALS and Huntington's disease. We made induced pluripotent stem cells that were gene-corrected. And we're making gene therapy vectors at the same time. These are the transfer vehicles that will transfer the genes that are needed for gene therapy. We're making AAV, adeno-associated viral vector, we're making lentiviral vector, and we're making retroviral vector. And on top of that, we're in the drug-making business, small molecule drug-making business, and we're making formulations of neuroactive steroids and drug and placebo formulations for the clinical trials. So I had a really tough decision to make. What am I going to weigh? No, that's not what it is. Which of the trials would I like to present to you? Let's do this. Let's do iPS-derived gene-corrected keratinocytes for the treatment of epidermolysis bullosa. Reason being, because one of our graduate students from Sac State that was in my laboratory interning worked on this project, and it was his thesis project. So what is this disease? Recessive dystrophic epidermolysis bullosa is a skin blistering disease. It's incurable. It's something you get born with. COL7A1 is the gene that codes for type 7 collagen. You wouldn't appreciate that if you wouldn't have the disease, but if you have the disease, you very much appreciate it because it anchors the skin disease to the underlying tissue. And if you have this disease, the skin blisters off, just when you brush over it. The kids that have it lose their skin already during birth. And they're called the butterfly children because they're so fragile. Minor friction and trauma later on in life will also impair them, and they're constantly bandaged. You can see the foot is bandaged here from the all the way, and you can see the blisters happening all over the place. Luckily, there's a severity range, and not all children are totally severely affected, but many are. And the ones that are so severely affected often die after a short period of time. Our task was to develop an induced pluripotent stem cell therapy for recessive dystrophic epidermolysis bullosa, RDEB, short, I'm going to call it from RDEB from now on. And we received one of the first CERM disease team grants in conjunction with Stanford University. Stanford University did all the basic research on that. They also did some of the translational research, and they said, you have to now translate that into a clinically applicable product because we want to treat patients with that. Wow. So what did we have to do? So we took skin fibroblasts from patients with RDEB. So there you have the skin of the patients that have the disease. I thought, is this a good starting cell to make induced pluripotent stem cells? Well, there's two things to consider. First of all, it's the actual cell that we have to transform then into a skin stem cell, finally, that will make these gene-corrected keratinocytes that make dermal grafts. That might be a good idea because they're genetically pre-programmed to do that. However, who goes tanning? Who goes to the beach? Yeah, see that what you get when you are exposed to the sun too much? Sunburn, and what do you get later on when you get older? Skin cancer. Because what happens? UV penetrates the skin and actually causes some chromosomal rearrangements, possibly some DNA breaks and things like that. So we took the chance, let's say it that way, to take this particular skin cell from that particular patient and make an induced pluripotent stem cell. How did we do that? Well, remember, this was 5 years ago. And the only real way of making induced pluripotent stem cells was using an integrating vector. So this vector transfers four growth factors into the cell. These growth factors are present naturally when your cells develop in your embryonic state because because they can make all types of body cells when these factors are upregulated. So we generate a vector that expresses these factors, and we put that vector into these skin cells. But if we use an integrating vector, what's the drawback? It stays in the chromosome forever. We don't want that. We want to remove that vector. We only want it in there for the induction of pluripotency, and then we want to take it out. So what did we do? Stanford sent us a removable vector that could be excised. And that system that they used was this LOX-P system. It has two recognition sites, this vector on the right and the left side. So you have a LOX-P site here, and you have a LOX-P site over there. And then it integrates semi-randomly somewhere in the chromosome. What you can do is you can add an enzyme called Cre recombinase by using a plasmid and transfecting that transiently into a cell. And the Cre recombinase recognizes these LOX-P sites and cuts out what's in between. And see what remains is only two little scars, these LOX-P regions, but they're not coding for anything, they're not doing anything, they just sit there. And removed is the cassette that has these four reprogramming factors. After a few trials and after some error and lots of colony picking, we got to the point that we actually could do that. So there we are. And now we have a much bigger problem. We have these iPSCs. We have to pick individual colonies, we have to propagate them. What do we have to do with them? We have to gene correct them. Here comes the idea. Well, we don't want to randomly gene correct them, we want to cut out the gene that's bad, and we want to replace that with a fully functional gene that makes collagen 7A. How do we do that? We use homologous recombination. It's notoriously inefficient. And here comes our iPSC system that helps us out, because we only need possibly one properly corrected cell, and since it is pluripotent, it makes a lot and a lot and a lot of cells. We can actually use just one cell and repopulate the whole dish, the whole flask, and the whole culture system with clones from these particular corrected cells. And that's what we did. So here comes the corrected cell. We find that by just randomly picking these guys. And then we take one of these and test it. And after a few tests, we find out which one is actually the corrected cell. So it's many, many late night hours for graduate students to do that. And you know how that works. So finally, there's this one corrected cell that actually works. And we actually had 3. We were lucky. And these get propagated. Then you have this highly expanded gene-corrected iPSC at the end. What do? You make keratinocytes. Then you run these cells through a differentiation protocol. And if you're lucky, at the end, you make human keratinocytes. These are the skin stem cells from which you can make a dermal graft. And that's the dermal graft, and that you can put onto a lesion, and then you can graft a patient with that. And this all has to be done in the GMP facility under quality control and quality assurance. And Aaron went to work. Aaron Schwarzkopf used to be a CSUS student, and he came into my lab, and he became very busy with that particular project, as you can imagine. And what he did was he grew these patient iPSCs. These are actual pictures of the patient iPSCs, and then he was able to gene correct them and swap out the gene therapy vector. And this is what he got at the end. He ran it through the differentiation protocol. And what you can see here is the following. You have these cells. You can see all the nuclei, they're DAPI stained. This is the phase contrast of all these so-called keratinocytes. We didn't know if they were really keratinocytes. And then we came with a keratinocyte-specific marker, and this was not just only one marker. We have several markers for that, and I wanted to point out this particular one because it's bright and you can see it here on the screen. And on the higher magnification, you can actually see these particular cells lighting up with another keratinocyte marker. And then you can see the overlay between the DAPI nuclear stain and the K14, and you can see clearly that we have mainly gene-corrected keratinocytes here. So what we did was then we, of course, did all the quality control on the cells. We PCR'd them up. We wanted to make sure that the gene correction is really there and that the bulk of your cells are really gene-corrected. And finally, this went down to mice that actually had a defect very similar to the human defect, and guess what? The dermal grafts stuck to the mouse. So that means we had a skin graft that could be put onto an immunodeficient mouse with the same disease and was able to produce skin that didn't blister. Well, this was finally written up, and guess what? We got into Science Translational Medicine. It just came out, it's not too long ago. That was in November of 2014, and this is our whole project that we have been conducting here for the last 5 years practically, and we have been working with Stanford to do this. So if you want to read that, this is soon going to be posted, and you'll be able to find this article online. And I wanted to tell you that Aaron went on to medical school after that. So you don't have to stay in my laboratory. You actually can do something different afterwards after you finish such a complicated project. All right, so that's a real difficult project to do, but we mastered it. And we actually found a way to make induced pluripotent stem cells under GMP conditions with a lot of fiddling around with the conditions, trying to get all these reagents GMP grade, and even trying to find feeder cells that could be made GMP grade, which we were successful with. Maybe in the future we may not have to do that because we may get into a feeder-free process, and in the future this may be rolled up again if we get more funding to really do this. Okay, now here. This was not completely translated into a clinical application, what I told you. Almost there. We had interactions with the FDA. We had this pre-IND meeting. We knew exactly what we had to do to get it into the clinic. But this is the project here that actually went into the clinic. And now I want to tell you what this is all about. Bone marrow CD34 positive cells for the treatment of an eye disease, ischemic retina. People look at me and say, is he a bit How can he do this with bone marrow stem cells? Put them into the eye? Does he want to grow blood in the eye? Well, let's see. Here's the hypothesis. Bone marrow stem cells not only form blood cells, we found, and Dr. Nolte already for years has been showing that in her papers, that these stem cells have regenerative capacity. They can promote blood vessel growth. That's the most important aspect, is really blood vessel growth. How does that work? Well, these cells release factors. Being brought to an area where there is not enough blood flow, these factors will be released from these stem cells, and they will promote new blood vessel growth. That's a fact. So this was spun a little further by Dr. Susanna Park here at UC Davis, a very good ophthalmologist. She thought these cells may actually rescue photoreceptors by improving blood flow. So when you become blind, it is often due to the fact that there is not enough blood flow in the retina. What if you restored the blood flow? Could you maybe rescue some of these cells that are not quite dead yet? Well, hypothesis. Let's see if we can prove it. So what we did was we took patients' own bone marrow cells and we transplanted them. What's the advantage of patients' own bone marrow cells? This transplantation has been done many, many times before in medicine, and it has been known for decades. Well, there's no rejection. There's no tumor formation because it's your own cells. Well, what's new about this is they're being not used for the same process of regenerating the blood system, but they're being used for making maybe not so healthy cells healthier again. So in vitro and in vivo experiments were carried out to confirm the hypothesis, and we were able in an animal study to show that these human bone marrow stem cells, isolated stem cells, actually were safe. And they could be put into the eye of these animals, and there was no issue. And there was even seen an incorporation of these cells along the blood vessels in the eye of the animal. If that actually really did anything to improve the disease was not quite clear. But anyway, we applied for an investigational new drug that we submitted to the FDA, and we said we want to test the safety of these applications of bone marrow stem cells in blindness. Primary endpoint, safety. We want to make sure it's safe to put them into the eye. Secondary endpoint, efficacy. We didn't expect much, but at least we wanted to try it, and the FDA said, okay, you can try it. So how are we doing that? We find an appropriate patient with the appropriate blindness, and I'm going to show you in a moment what type of blindness we're talking about. There's an eye exam and more exams going on at UC Davis. Then there's an outpatient procedure if the patient qualifies for the clinical trial. And a bone marrow aspirate is taken by a very experienced physician that does this all the time, because we need a good amount of bone marrow, but we don't want to put the patient under general anesthesia. We want to do this under local anesthesia, so this has to be done just right. And then this happens. It comes to the GMP facility. There, we do a GMP-grade magnetic bead selection. This is how we pull out the CD34 positive bone marrow stem cells. We label them with magnetic beads, and then we run them through a column. And that column with a magnet holds back the labeled cells while all the other cells run through. And at the end, we elute the bound cells, and then we have our pure CD34 positive bone marrow stem cells. Finally, we do quality control and quality assurance on that. We want to make sure that we have no contamination, that everything is safe, and then we want to know how many bone marrow stem cells we really caught. And finally, we fill that into a syringe, and now comes the trick. We're taking out a good chunk of bone marrow, and then into the eye goes 100 microliters. Now, we have the task in the GMP facility to make sure that we get all the bone marrow stem cells into 100 microliters into a tiny syringe so we can actually inject that into the patient's eye. And this all needs to start at 8 a.m. and finishes at 5 p.m. back in the patient without quality control. Did we do that? We did. Here's the paper. Exactly. We already treated 6 patients. So that's a UC Davis investigator-initiated IND. And we were the first one in this country to actually do that and then pull it off and treat 6 patients. And I want to go through these patients because it's very interesting what they actually had. So CRAO means combined central retinal artery and vein occlusion. That means that there's very little blood flow into the eye, into the retina, because the vein and the artery are occluded. It doesn't carry enough blood. That's the first patient. Then we had Stargardt's disease, which is, you know what macular degeneration is? That's photoreceptor degeneration in the eye. And we have AMD, age-related macular degeneration. But there's a younger onset too, and this is Stargardt's disease. So we had 2 patients with that disease. Then we had 2 patients with age-related macular degeneration, and 1 with retinitis pigmentosa, which is an inherited disease of the photoreceptors. So what we did is we got the bone marrow, we got sufficient cells out of the bone marrow, we did appropriate quality control, we had no contamination, and we got an appropriate number of cells back into the patient by 5 p.m. with wonderful viability. So look at the first patient. Who has been to the eye doctor? A lot of people, and you read what? An eye chart, and this is how you determine your vision. And there's a line, and if you can read 20 and 20, then you have 20 and 20 vision by line number, okay? That's on the eye chart. And that particular patient, number 1, had a vision of 2800, which is legally blind. You can't see the fingers in front of your eye. That patient had the combined central retinal artery and vein occlusion. And guess what? After the stem cell injection, the vision improved to 20/40. And that's remarkable. And that was sustained after 6 months to 20/63. There may not even be a statistical significance in between the 2 readings because one may be biased a little bit and whatever, but there is clearly an enormous improvement from 2800 when you can't do anything to 20/40 when you can do practically everything. Because I have 20/80 vision when I take my glasses off, and I can still see you. So, Stargardt's disease patients. Well, yeah, let's have a look at them. We did have a little bit of an improvement. You also see underneath a number in parentheses that says 65 and 55 on patient 1 in the follow-up, in the red and the green. That means improvement in letters, reading. There was an improvement in 9 letters and 7 letters and 11 and 10. So there is some improvement, even with young onset macular degeneration. Now look at that. Then we have AMD, age-related macular degeneration, in this patient that was 76 years old. The initial vision acuity was 2200. And guess what? What happened? We got it to 20/80 sustained in age-related macular degeneration. That's enormous. So we have 2 patients now with 2 different types of diseases. So this was not a single incident. No, we could repeat it, but we could repeat it even in a patient with a different type of disease. Then we had retinitis pigmentosa. We did have quite some improvement after the first injection. There was only 1 injection after the first follow-up. And there was very little improvement in AMD of the second patient. But this was an 85-year-old patient, and we may not have gotten the best possible bone marrow from that particular patient. But what you can see is we tried it through all ages. And it seems to work with a younger person, and it seems to work with an older person. And it's clearly undisputable. Let me show you even more. Here is the retina fundus photography that we did on the first patient. This is subject number 1 with this enormous improvement. So what do we see here? On the left side, where the arrow points, you can see all these little specks. Do you see that? This is bleeding into the retina. So these are hematomas, little hematomas. When you get bumped, you get a hematoma. Well, you have a hematoma in the retina that looks like that. And in picture number B, you can see that this is almost fully resolved after stem cell injection. There's no doubt about it, okay? This is not just reading letters, this is real data. Then finally, you can see in the second one, Microaneurysms, this is little blown up parts of the arteries. And retinal ischemia also, this is areas where there's really very little blood flow, because you can see that actually the white gets a little lighter here when you look at that right underneath the arrow. You can see that there's ischemia. And this is quite improved in picture number D after treatment. So we have the data here. So this was published again and came out in January in IVUS, and you can also look this up and read it in more detail. But here we can show you that with an investigator-initiated stem cell application, that's the only one currently in the United States, we can make a difference and can give clinical benefit to patients with blindness. And that's even if this one thing would have been done in the GMP facility only, it would be a great benefit to patients. We're working on amendments to our IND, making it easier for patients to enroll, and hopefully we'll get some funding too to be able to enroll more patients. So, how did we do all of that? I made it so simple and easy to get to the whole regulatory process. This is why I wanted to bring this in the middle of the talk, but now I want to show you what we're doing to assure that this is scientifically sound and that it's actually safe for the patient. We go through our Stem Cell Research Oversight Committee, SCRO. First, they have to approve the ethical use of stem cells. If we do gene therapy, like we did for our epidermolysis bullosa project, we have to go to the NIH Recombinant DNA Advisory Committee, or the RAC. This is where they pin you up on this thing and then they stretch you. This is how it feels when you go there, because you have to appear in person. And then your peers question you. And then you have to answer their questions. And this can be a bit daunting, but we got through that. Then you come to me, because I'm the vice chair of the Institutional Biosafety Committee. But I can't review my own protocols, of course, but if other people come to me, I'm able to do that. And this is the extension of the NIH RAC at the institution. Finally, it has to go to the Institutional Review Board, where the safety and the ethical component of this particular clinical trial is being looked at so you don't harm any patients. And finally, at the end, you end up at the Food and Drug Administration. That is the final authority that will approve or not approve your clinical trial. After that, you can initiate a phase 1 clinical trial, which is a safety study. And this is the timeline. So you start out at day 0 with the SCRO, and then you go to the RAC, you go to the IBC, you go to the IRB, and then finally at the end you end up at the FDA. And this is what you need to do. You need to produce preclinical data according to good laboratory practice. That means you have lab books where everything is written down appropriately. Students, do you do that? I see some faces here that have, okay. All right, so from now on you do it. And if you write it on a glove, you keep it. You understand that? Because that's where you want to go back to, that's your source document. I know all of that. I did it. Okay. So use cell lines that can be completely traced and characterized. Yes, if you don't know what you have and do you want to put that into a patient, people have tried that before. They really did. And it failed miserably because they harmed patients. And then finally have detailed information on reagents. You don't want to throw out your spec sheet that you have with the reagents. You want to keep it. What are suitable preclinical data for a phase 1 clinical trial? Safety data, of course, because it is a safety study in an in vitro and in vivo model, in the relevant animal model, but you will never get away without any efficacy data nowadays. So the FDA really has made a big effort to tell people you need to have efficacy data and show that you're actually doing something that could benefit a patient. Finally, the product should be safe and should be able to produce the therapeutic effects in human studies that you would like to have. So when this is all in place, you write an IND, an investigational drug application. You need to consider good clinical practice and good manufacturing practice. You define the endpoints of your study. So what was the endpoint in the retina study? What do you think? Safety. So we wanted to look at the safety parameters. We wanted to see if we did anything to the eye and how did we do this appropriately. In science, we need what we call controls, don't we? That's number one. If you can't control it, you don't know what you're doing. In our eye study, what do you think was the control? The non-injected eye. So we left one alone and the other one got injected, and you could always compare to the non-injected eye. It's simple, isn't it? But it's a wonderful control. If this works, you can always re-inject the other eye. See that later on. Finally, I wanted to give you a hint. If you do a gene therapy study, there's a 15-year follow-up of the patient. And this has something to do with the bad things that happen in the beginning of gene therapy when a patient was killed, unfortunately, from a bad gene therapy experiment. The FDA has given in on this 15-year follow-up a little bit, and it's negotiable, but still keep in mind that it takes 15 years of follow-up for a gene therapy clinical trial. Then you request a pre-IND meeting with the FDA. You submit a short write-up of what you're planning to do. And finally, if you had that, you submit the IND. And here comes the fun thing. The FDA will allow you to go ahead with your clinical trial if they don't say anything within 4 weeks. But this usually never happens. Before the 4 weeks are up, probably an hour before the time is up, they will call and say, "You're on clinical hold." That's all they're going to say, because they did not have the time to review your IND in depth, you know. And then you will have to actually go back to the FDA and negotiate that. After 4 weeks, you get a final thing, and there's only 2 points in there that they actually point out that you maybe have overlooked. You didn't send this and you didn't send that. Where is it, okay? And then you send that in, and finally you'll be taken off clinical hold. And this is normally how it looks like. This is me, okay? I think so, at least. This is after 14 months. This is how we look after 14 months, don't we? And finally, this is when the clinical hold happens. But then, after 18 months, you got FDA approval. And finally, you're allowed to go ahead with your gene therapy manufacturing, cellular manufacturing, or other manufacturing. And this is how we do it in the GMP facility. It's not complete for me to give a talk here if I don't show you what we're doing in the GMP facility with a few little improvements that we have done. As you can see, our students, these are Sac State students, of course, looking at the microscope. And I was taking the photograph, and they were behaving because they were so afraid of me that they could do something wrong here. See how delicately they actually touched the flask, and I love that. And then here you can see some MSCs that look really good in the microscope photograph. And then these go into bigger flasks, and they go into even bigger multi-layer flasks. And finally, you have a product that you can put into a patient after you do quality control and quality assurance on it. However, that's often not good enough for the larger scale that we need to target for cellular therapies. So there's another Sac State student here holding a 10-layer cell factory. This is a bit bigger than these 3-layer factories. And there you have liters and liters of culture media, and we have 1620 of these in our incubators. You can see the Banker incubators in the background where these go in, and you can manipulate them still in the biosafety cabinet manually. But it's quite treacherous to do that. So you sit there for hours and hours and hours, and when you come out of there, you're wiped. So there's a different system that we're applying currently, which is a closed system hollow fiber bioreactor, which you can see on the right side. The cartridge contains millions of little hollow fibers. So you culture your cells inside the hollow fiber, which is semi-permeable, allows for media to percolate through the fiber. On the inside and also from the outside, the cells attach, like they would attach to a tissue culture flask, and you can grow them in there. The nice thing about the system is it is functionally closed. You don't have to work in a biosafety cabinet. You can put this instrument on the tabletop, and it grows your cells sterilely without antibiotics added, and you, at the end, get about 2 billion cells from such a cartridge. And you need a 40-layer cell factory or 4 10-layer cell factories to achieve the same thing as you would achieve in this small hollow fiber bioreactor. And where is this? That's in the Institute for Regenerative Cures. It's a 6,000 square foot facility, and as I say, it's still working like it did in 2010. We have 6 manufacturing labs, 3 intermediate labs, and we opened in 3 of 2010. Okay, little excursion into clean rooms. A few people were here before and heard me talk, but a lot of people have not heard that. What is a GMP facility? It's a clean room facility where you have very few particles in the air because you need to have a controlled environment. You need to control the temperature, the humidity, and you need to control the amount of particles. Why particles? Because in the air here we have 35 million particles per cubic foot, and we don't even know it. And they contain what? Spores, fungus, anything you can think of could fly into a culture. And a culture does not have an immune system. And we have to keep these things away from a culture. Therefore, we need to make the air really clean. So we have two classifications inside the clean room, a class 100,000, ISO 8 these days, less than 100,000 particles per cubic foot, and class 10,000, less than 10,000 particles per cubic foot. But that would not be good enough to do tissue culture. No, then you need to go into a biosafety cabinet. You know what that is? You work in it, don't you? With your cultures, that's where you put your cultures in. That's where you do all of your sterile manipulation. That's actually a class 100 clean room. Yep, and you have that in a dirty room, and then you go into class 100. It means you can bring some contamination. This is why you make a clean room, class 10,000, in which you put a class 100 biosafety cabinets, and then you clean. The class 100 biosafety cabinet is the most important lifesaver that you will encounter in your career if you choose biology and cell culture. So, this is how the GMP facility looks. I'm going to show you here with my pointer as good as I can. This is a gowning area from which you go into an intermediate entry area. Then you go into manufacturing rooms, and then you exit into an intermediate exit area from which you go into a degowning area. Simple, isn't it? This is actually repeated symmetrically on the other side. What's the advantage of such a layout? Look at that. You go from the gowning to the intermediate to the manufacturing into the intermediate exit into the degowning. Allows you one-way personnel, product, and waste flow. No product can be cross-contaminated with another product because you go in from one side and you exit on the other side. You never bring anything back. And therefore, you can manufacture 6 different products at the same time without interfering with each other. Now you know why we can make so many products in the GMP facility safely without any issues, without the FDA saying a thing about it. And this design I came up with about 20 years ago already. And it has proven really, really great, and a lot of people have looked at that design and have implemented that in other industrial GMP facilities. That's not all. We have to make gene therapy vector, and we also have a radioactive hot cell in there to make PET reagents, vials with radioactive material that can be brought over to the hospital, injected into the patient, and then they snap a photo, and then the radioactivity goes away because the half-life is so short. However, they could possibly be aerosolized. They could be in the air. They could be just blown out. So therefore, the FDA requires us to have pressure sinks in areas with aerosolizable material for gene therapy vector, or even fluorescent activated cell sorting. We have a cell sorter in there that also creates aerosols, and finally for the hot cell. And what we did was we actually produced a GMP facility that had switchable room pressurization the way you need it. Negative pressure for aerosolizable material manufacturing, positive pressure for the manufacturing of cells. And how can you keep a negative pressure room where stuff gets sucked in clean? Huh? Yeah, that's what I had to solve. We declare every manufacturing room a biosafety cabinet. This is why I say the biosafety cabinet saves you. You create laminar airflow from the ceiling to the bottom. It turns over on the bottom, goes up into the ceiling, gets filtered, and comes down clean. This is how you can actually create a negative pressure area without contamination. We have proven it now for the last 5 years. We put down touch and settling plates. We know how many colonies we can actually grow on these agar plates, and we don't have any. And this has been the case for the last 5 years. So all mechanical space, there's a space above the GMP facility where all mechanical equipment is in. Even the light fixtures are sealed in, because people forget when you open up the light fixtures, the first thing that falls out is fungus, aspergillus, and then you get it in your facility forever. So I engineered it so they're sealed in, and you change your lamps from above. We have individual air handlers for each room, and we have a computerized facilities management system that controls all air pressurizations and sends me alarms in the middle of the night, and I have to get up at 2 o'clock and fix something. And I don't even have anybody that actually comes in at 2 o'clock because sometimes they're out of town and I'm the only one on call. Well, that's what you have to do. And this is how it looks. This is one of our manufacturing rooms, and you have 2 biosafety cabinets in there. You can see up here on the ceiling is the light fixtures that are completely sealed in. There's no ceiling ties. This is all a tight area. We have pass-through here's. And then besides the electronic management, we can also control our air pressurization manually, and we go in every day and record pressurization, temperatures, humidity. This is our fluorescent activated fax sorter in the fluorescent activated sorter in a biosafety cabinet. So we built a specially designed GMP sorter where we can sort out cells under GMP conditions, the only one currently available. And this is our GMP grade hot cell, where we can make our PET reagents. And finally, this is my last slide. It's most important, is the acknowledgement. I'm just a director of this. So I sit in the office and I freak out, and try to get grants and get contracts and things like that, and talk to other colleagues of mine. But really, the work is being done by all these people. And we've had all these students from Sac State. Aaron Schwarzkopf, who is now at medical school, he did the IPSC project. Brian, who is our lab manager, he was here. Brian Fury was a CSUS student, okay? Steve Tobin, he is now a PhD student in Berlin and is doing very well. And all the other people, Jonathan Shu, our QA manager, Kasha, she grows all of our cells. Tia, she grows more cells. And Bill used to be our first employee in the GMP facility, really, with Steve, and now is doing all the regulatory affairs. So they have done quite well. Finally, we have a QC testing laboratory. John Walker runs the QC testing lab. It's independent of the GMP facility, and he used to go to CSUS too and got his degree here. And Kevin Yanamura is also a clinical laboratory scientist, to make sure that I say it correctly. Finally, I have to thank the UC Davis Stem Cell Program. It's Susanna Park, who did this whole thing on her own with the IND for the CD34 cells, and I helped her to get the IND through, of course. Meredith Abedi, who is taking the bone marrow from the patients. Suzanne, who helped us with all of the initial Clinimax runs, and Geraldine, who was actually former Miltenyi, and helped us to get the whole thing rolling. And then Jan, of course, who supports the whole laboratory, because without her, nothing would happen. And of course, all the other NOLTA lab members. Finally, Stanford, we must not overlook that, because Tony Oro was the one that really wrote the grant and was responsible for all the work at Stanford, and Al Lane, and all, of course, all additional authors and collaborators that we have to get this whole facility going. And I wanted to thank you very much for your attention, and now I'm ready for questions. Thank you.
Small question. How much does your GMP practice, or do you think your GMP practice help to formulate FDA standards? I know they have to approve your final procedures, but some of that stuff, I'm sure they don't have things worked out.
Is this the first question?
Yeah, first question. Second question is are we working toward an international GMP standard?
So, question number 1 was, how much standards did we set in the GMP facility over the years with the FDA? We have set a lot of standards. 21 CFR 210 and 11 is vague. And of course, when you build a GMP facility for cellular manufacturing, vector manufacturing, all of that, it's always going to be new territory. So what we do is we have a Type C pre-facilities meeting with the FDA where we submit our plans, what we want to do with the facility, how the facility is constructed, how it's going to operate to the FDA. And they approve that or have questions. And every single time we submitted such a plan to the FDA, later on in a guidance document, something was written in there that came out of that particular pre-facilities meeting, which was very interesting. For instance, the negative pressure is a very good application. In the past, a negative pressure of 0.01 inches of water column was actually the norm, and that's almost impossible to maintain at negative pressure. So I said, this has to be minus 0.05, and you have to have plus minus 1 or 2 inches tolerance because it has always needs to be negative. It can never go into 0. And they actually accepted that, and this is now the standard for that. International. So what we did was, I have tried to talk to the Paul-Ehrlich Institute in Germany about that. And with a collaborator in Berlin, currently we are talking to them about vector manufacturing, and we are applying, and there's not a lot of vector manufacturing going on, for instance, in Germany. And so they're looking to the United States, seeing what we're doing with vector manufacturing and seeing what standards need to be applied also in Europe. So there is kind of a harmonization talk going on, but full harmonization is never going to happen, I can tell you that. But there's going to be some common ground that we will be able to work with.
Additional questions?
Don't be shy. You can ask me something. Oh, there, see? Yell it and I'll repeat it.
So for your treatment for epidermolysis bullosa, the science regarding the background of it, what was the endpoint for that? Since the skin regenerates so quickly, a dermal patch, for example, would only work for so long, right? You'd have to replace that constantly?
I don't think so. Our hypothesis was a little different, and I'll explain to you why. So first of all, the skin does regenerate, yes, but if you use keratinocytes, they are always regenerating themselves. So they're skin stem cells, they're making more of themselves, and they differentiate then into other cells of the skin that are going to be shed. So if you make really keratinocytes that can regenerate, you may actually have made a patch that will last for a very long time. I can't tell you exactly how long, but it should last for a prolonged period of time. And also the patch may actually have a selective survival advantage. While the other cells right next to it may blister off, the patch may actually grow. And if we're lucky, it may actually expand. How much it may expand, I don't know. And these two things were taken into account. But really, for this clinical trial, all we wanted to see, and this is what we told the FDA, is we transplant one patch and see if it's going to work. Reason being, there could always be the risk that cancer forms, the teratoma forms, or something forms, and we want to watch this one patch and want to make sure that this one patch actually is safe. The nice thing about one patch is you can excise it in case something goes wrong. Here.
You're close enough. I'm sure he can hear you.
Just yell it out.
I know we're working on right now the Xeno or nanoforging. And I heard you mention making GMP mouse feeder cells. Can you tell me about those cells and what makes them different from?
Yeah. So, first of all, in the beginning, we were not able to grow any iPSCs on anything but mouse feeders. We tried everything under the sun, and please remember that was 5 years ago that we started out with that. The culture systems were not as developed as they are now. So all we could do at that particular time was try and make GMP-grade mouse feeder cells. The FDA had no issue with us using mouse feeder cells at that particular time as long as the mouse cells were tested appropriately for adventitious viruses and the absence of other contaminants. That would have gone through a huge amount of testing, but if we had such cells, they could have been used for limited numbers of patient cell productions. So that was not an issue. It's always advantageous, as I say. That's the FDA language that comes out of me right now. It would be advantageous if you developed a system that would be completely xeno-free and would contain totally defined products and reagents for manufacturing. Yeah, that would be advantageous, but it's impossible currently. So, therefore, the FDA has always considered to use things that make a product efficacious and safe in vivo. That means in a human application, which means that we have to test any and every possible contaminant that's in there or any adventitious virus. Why does the FDA want to get rid of xenoproducts? Because any xenoproduct in a patient could cause an immune reaction, of course, but so far, if you do one transplant, this hasn't been seen, really, to that degree. But it would be advantageous, and we're working towards it, of course, but it's not there yet, and if we don't want to keep these products that could be very beneficial from patients, then we have to continue with what we have right now and weigh the risk. There's always a balance, okay? Are you going to be doing something good, or are you going to create anything that could harm somebody? And you always have to be on the side where you do something good, and then you put your improvement in later on, which is called the C, the current good manufacturing practice.
Thank you.
Did you have a question? Any additional? Oh, here we are.
You mentioned before that before going on to clinical trials, you have to test it, in relevant animal models. And what are these relevant animal models? Are they immunocompromised mice, or how do you determine a relevant animal model?
Outstanding question. So there's both animal models available. Some animal models are just not available as immunocompromised models. What is an immunocompromised animal? This would be a mouse that does not have an immune system, for instance, in which you can put human cells that will grow, and the mouse does not recognize these cells as foreign and will accept them as the mouse's own cells. And then, for instance, we do this all the time when we reconstitute a human immune system in a mouse. And then we can test all of the human cells that would arise in a mouse like we would do in the human. But if this is not possible, you have to do whatever you can to get to the relevant animal model. You have to take the animal cells, you have to gene correct them and put them into the animal model, and then demonstrate with these cells that you can actually cure the disease in the animal. If that translates into the human model perfectly, is an unknown. Therefore, it would be fantastic if we had everything in the immunodeficient background and we could use these human cells and put them into the animals, but often that's not possible.
Back to your bone marrow CD34 stem cells, you mentioned that you were able to treat 6 patients, but why was the clinical trial pool so small?
Because you didn't give us enough money. That's the reason. Okay? So the issue was it was unfunded. So it was just donors and all kinds of donations, and the university pitched in to actually fund the clinical trial. Good question. Thank you.
Did I understand correctly that you do steroid manufacturing as well? And
I do what?
You do steroids. Steroid small molecules. You said like at some point you...
What we do is formulations of small molecules.
A formulation, yeah. Drug product manufacturing.
Yes. Mm-hmm.
So if you do, so can you do high potent cytotoxic small molecules as well? And if you do, what kind of OELs are there?
We don't do that currently, because what we have is small molecules that we do, and we currently don't do any toxins or anything.
How do you choose the projects you do, or how have you chosen them? And is there anything in the future that you're excited about coming down the pipeline?
Yes, so there's a little hierarchy. First of all, you have grants, of course, and if you're grant funded, then you do the project that you're funded for. Second of all, you have your customers. And there's, of course, the customers that are in line. You take the customers, you see if the project can be done, and you do your project with the customers. So what we developed was a 2-tiered system, investigators from the inside and investigators from the outside, and we developed GMP facility rates. And what we can do is we can offer these rates to inside and outside customers, and we can relatively easily set up through contracts a manufacturing for small companies which are in need of GMP manufacturing. So 50% of my business is practically grants, and the other 50% comes from outside companies that need to have a product manufactured. And the diversity comes in with my customers. It's really interesting. So we started out with Stanford. That was my first project, really. And then suddenly the drug formulation came in because they couldn't be manufactured anywhere else. So I had to come up with a system to do that. So on the fly, I developed all kinds of things. And then came the vector manufacturing. I didn't know how to do AAV vectors, so I had to learn it. How about that? You know? So all that came in over time. Okay, so this is also through a company. I can't tell you too much because I'm under non-disclosure, but I can tell you what has been published on their website. So when you take dermal fat, okay, and dermal tissue, you can take the whole tissue in small chunks, and you could put the gene therapy vector into the whole tissue, and all cells in there get exposed to the gene therapy vector, and some of them will actually take it up. And then they will release the gene product over a prolonged period of time. And for instance, this can be done to replace the injections that you need to get when you are on hemodialysis and you're erythropoietin deficient, because you get injections every day practically to replace the erythropoietin. You could have this for a few months and they're working properly without you getting injected. It's quite a benefit to the patient.
We have time for maybe one or two more questions.
When you mentioned about the blister and skin disease, you would had a little bit doubt about using the keratinocyte from the disease patient. Would you consider using the bone marrow MSC to make it into iPS cell instead of using the diseases keratinocyte?
So this was a long discussion, of course, that we had overall. Which cells would be the best cells to use to treat the patients with, okay? So we decided on the keratinocytes because we had keratinocyte lines already available. And some work had been done with these keratinocyte lines. And also the keratinocytes that we had from these patients could easily be further manipulated, okay? So we had fibroblasts and we had keratinocytes. And we had keratinocyte lines that could be propagated very well. And we thought, if we fail, we can make more keratinocytes and we can propagate them better and we can use these keratinocytes for more. This is why we actually used the keratinocyte line, but it would have been no different, I think, if we used fibroblasts, fresh fibroblasts from the patients. And we were actually considering to take a punch biopsy, take skin fibroblasts, and use that for a fresh attempt for gene correction.
Okay, you'll be our last question.
So what would you say is your largest hurdle to creating the GMP facility? Was it more of like a scientific side, or would you say it's the business aspect of it?
Business aspect, absolutely. The issue of academic GMP facilities is the following. You get the initial money to build the academic GMP facility, and afterwards you have to run it. Where do you get your customers, okay? So we were lucky that we had CERM, and that we got CERM funding to start out with. But we also, during the time, developed a reputation, because Gerald and Annette helped us with the rates. Yep, I have to say that. That was the best business decision we ever made. And we developed a rate for outside companies that could come and could have their products manufactured in our GMP facility. And that keeps us busy. I think we would be out of business if we wouldn't have that, to be honest. So that's a very good model. And also, you want to engineer the facilities so you have the least amount of operating costs. And this was done with this particular facility. Thank you.